And for one thing, I want to make sure that I get through all of the slides that I have and give you all of the materials that I wanted to cover today. Rodeo had asked me to talk about our mechanical ventilation guideline, which was published within the last few months. And so I'm going to do that today, but I'm going to take the opportunity to talk a little bit about guideline development too. The reason for doing that is there are a couple of reasons for doing that. One is because I think that if I were to just present to you the recommendations from the guideline, that would get fairly boring very quickly. So I'm going to try to make that a little bit more interesting. I've included some questions for all of you. But I also want to tell you a little bit of a story, to go through the story of how this guideline was developed. And in doing so, to give you an idea about how guidelines like this are developed. I think that this is an important element of your education for those of you who are fellows, because understanding how they're developed and what exactly the recommendations mean is important. You'll be using these guidelines more and more often throughout your careers, because I think this is an area that is going to increase in importance in all facets of medical practice. And so I'm going to talk a little bit about that too. The formatting of the slides is not meant to be propaganda or to be anti-Henry Ford or anything like that. I just didn't want to reformat some of my slides, and so I left them all in this template of chest. So that's what that's all about. And I have several disclosures here, and I have to tell Rodeo that the reason that none of them are on the thing that you just had me sign was because the document was 12 months, and these are either before 12 months or they are going to occur in the future, and so there are differences between those two things.
Yeah.
Okay. What we're going to do today is to talk a little bit about evidence-based guidelines so that you have some understanding about what those documents mean. We're going to talk about several of the recommendations in a recent mechanical ventilation guideline. I've put 3 of the recommendations, or 3 of the learning points here, but there are actually 6.5 recommendations in the new guideline, and we're going to talk about each one of those. So I'm going to start by talking a little bit about evidence-based medicine with a quote inevitably from William Osler, who a century ago talked to us about how medicine was both an art and a science. The practice of medicine is an art based on science. Medicine is a science of uncertainty and an art of probability. And I think that's as true today as it was 100 years ago. Archie Cochrane is a doctor from the mid-20th century who started developing the Cochrane Collaboration, and a quote from him would be, "You should randomize till it hurts." So, what does this have to do with either guidelines or mechanical ventilation? This is Mrs. Winslow's soothing syrup, and these boxes are really picturesque, right? These were developed in 1849 in Bangor, Maine, and what you did with this syrup was to use it for teething, for infants who are teething, because it would relieve their discomfort and quiet the infant. And if you look at the active ingredients, which included morphine, it was probably a fairly effective remedy, but maybe not one that we would use today in terms of risk-benefit management. This is a more prosaic treatment, the health jolting chair, and you will see that this is efficient exercise to support essential nutritive organs. Perhaps not something we might use today. So these were patent remedies, which were common in the 19th centuries. And patent remedies are defined as a commercial product that's advertised for certain purposes without regard to its effectiveness and, I should say, safety, given the first one. Well, in the 20th century, our perspective changed, and we sought scientifically based treatment. And here on the left hand, the photo is Sir Alexander Fleming and his first patient who was given penicillin, the chemical structure of penicillin, and there, of course, at the bottom is a petri dish showing a mold colony that is elaborating penicillin and not allowing organisms to grow in the adjacent vicinity. The idea here is that the development of penicillin as a treatment was scientifically based. And so this became a very important 20th century idea that we moved from patent remedies, which were common in the 19th, to scientifically based treatments in the 20th century. This is a device which I used all of the time when I worked in the ICU as a resident. In fact, I don't think a day went by when I wasn't putting one of these in. Now it's hard to find residents or even fellows who are credentialed to put them in. We based the use of this on scientific principles. Here's the waveforms that you can copy or imagine when you place a Swan-Ganz catheter. This is, by the way, there's a couple of references here that if you're interested in history and also if you're interested in some of the basis of what we do, they're worth looking at. This article by Swan and Ganz is important and useful and interesting to look at, and the one by Forrester as well. This is an examination, a comparison of the wedge pressure, and the central venous pressure is measured by a catheter on the right side. And you can see, very interestingly, in the group of patients they looked at, there was no correlation between the two measurements. And they used changes in either volume loading or diuresis and compared changes in CVP and wedge pressure, showing no correlation. And then in what I thought was a pretty elegant study from 50 years ago nearly, they looked at ventricular filling pressures and evidence of congestion on the chest radiograph. And they demonstrated that the wedge pressure was able to distinguish in their group of patients by using the wedge pressure between those that had and did not have vascular congestion on X-ray, whereas the CVP did less of a good job. And so we started using these catheters all of the time to estimate preload in the heart. And we used this catheter, in a sense, based on scientific principles. There was a scientific principle behind this intervention. The problem is, when we more carefully studied this in later times, this is an article from 2003, we found that the use of these catheters made no difference in our ICUs and patient outcomes. So even though the intervention was scientifically based, even though we thought as doctors we had a better idea of what was going on, when you looked at a population of ICU patients, you couldn't show a difference in outcome. And so the next step really in what we're doing as doctors when we look at evidence is to take scientific-based interventions and say, in the 21st century, you know what, that's not enough. We need to know that this intervention based on science actually improves an outcome for the patient. Now, Dr. Cochrane in '71 published a pamphlet, Effectiveness and Efficiency. And what he recognized is, yes, randomized controlled studies looking at outcomes were important, but the problem that all of you know, is that sometimes randomized controlled studies are different one from the other, that sometimes they look at the same outcomes and find different results, and sometimes they find results that perhaps are consistent across trials but don't reach significance in a given trial. And what Dr. Cochrane suggested is that we need to develop some sort of tool that summarizes the information for the questions that we have and to look more comprehensively at these questions instead of looking at one trial. And so evidence-based guidelines are an effort to fill that role, to take the scientifically based interventions to look at outcomes, but to provide summative data across the available literature to guide us in our practice. These are the Institute of Medicine standards for developing trustworthy guidelines. What does that mean? The Institute of Medicine has established basically a scientific framework for doing guidelines. And these are the principles. And if you get this pamphlet, you'll find out that each one of these principles that I've listed for you is actually the title of a chapter. So there is very extensive knowledge and background behind the development of these principles. Calling something a guideline by itself does not mean that it's an effective guideline or tool. For example, if you go to look at the GOLD guideline, which most of the pulmonary fellows are familiar with, and you try to apply the Institute of Medicine standards to that document, you will find that the GOLD guideline falls short in many aspects. So this is a way purported by the Institute of Medicine to develop guidelines in a scientific way so that they make sense and that they're useful documents and that they have some reproducibility and credibility. So my first question is not about the mechanical ventilation guideline, but about guideline in general. So the question, I'm going to read this and then we're going to open it up for your responses. Which of the following is true about evidence-based guidelines? Evidence-based guidelines are developed to direct pay-for-performance measures. Evidence-based guidelines are developed to provide evidence-based recommendations to inform clinical practice. Only randomized controlled trials can be included in evidence-based guidelines. Evidence-based guidelines are developed to direct the development of public policy. Pick the best answer. So do I need to do anything for the polling, or does it just automatically appear?
Just click on the next slide.
Next slide. All right. So everybody picked the right answer. The problem with the right answer, the problem with the wrong answers is that some of them are relevant to our discussion today. And although we try to develop guidelines to guide practice, pay-per-performance, that they're used to direct for performance measures. And they are also used to direct public policy at times in terms of Medicare and governmental payment. And so those things are important. You guys are correct in thinking that randomized controlled studies are not the only thing that you can include in an evidence-based guideline. You can include all types of studies, but the strength of the evidence has an effect on how strong your recommendation can be. So this is a second evidence-based guideline question, a little wordy. I'll read it for you. Dr. L is a candidate for a position on a panel to develop an evidence-based guideline concerning lung transplantation. Just picked it out of the air. Could be anything. She has received $16,000 from Company A, $19,500 from Company B, and $12,000 from C for Speakers Bureau activities for immunosuppressive drugs last year. She has also expressed strong opinions in an article in the national press concerning a non-pharmacological treatment, and she hasn't been compensated for making that statement. So which of the following is true? Because Dr. L declared her conflicts of interest, she should be a panelist. Dr. L's Speakers Bureau's activities represent a conflict of interest, but her non-compensated public statements do not. If Dr. L is a panelist, the sponsoring societies risk legal action and congressional inquiry. Dr. L may be a panelist only if she divests herself of her activities and promises not to engage in them. Which is the right answer? Ten seconds gone by. The answer, unfortunately, the correct answer didn't do well. So the real answer, and the most important one, is that the sponsoring societies of this guideline would risk legal action and congressional inquiry. The Infectious Disease Society of America was actually brought into court by the Attorney General of Connecticut, and the reason for that had to do with a consumer advocacy group who challenged the guideline on Lyme disease that they produced. And the basic tenet for the suit was that the guideline experts were basically in the back pocket of the pharmaceutical companies, and their recommendations didn't include recommendations on chronic Lyme disease. And the ISDA had to revise their recommendations, and their suit cost them well into 7 figures in order to defend themselves. One of the cardiology societies, the American College of Cardiology, was called in front of Senator Grassley's committee on the Hill because of a guideline that they published where their experts were highly conflicted because of Speakers Bureau's activities. So this is not a minor point. Medical societies risk significant problems when this happens. Dr. L declared a conflict of interest. She should be a panelist. The answer is probably not. There are different ways to work with somebody who has conflicts, but simply declaring them is probably insufficient when someone has conflicts that are of this magnitude. The promise of divestiture is a necessary promise for anyone participating in a panel that meets Institute of Medicine standards, but it's not sufficient if past activities were important. And more folks are looking at things that we call intellectual conflict of interest. There are several medical societies that look very carefully at intellectual conflicts of interest in public statements in terms of panelists. What most panels try to do is that they try to impanel members who have different perspectives so that you get a balance. But this is something that's being looked at and is important. So conflict of interest in terms of guidelines, at least, is not a minor issue. It's a major issue, and folks have gotten into a lot of trouble for not paying attention to it. So the process of developing a guideline is extended, and I want to tell you a little bit about the guideline for mechanical ventilation. Early in 2000-2001, the College of Chest Physicians had a guideline on mechanical ventilation, and it was in that guideline that we first started talking about spontaneous breathing trials, and they were recommended, and some other things that you're used to doing very commonly. And I, at that time, read that guideline and thought it was a wonderful document. In fact, it was one of the things that got me interested in learning about guidelines and being involved in them. 2 or 3 years ago, I found out that there was some interest in the journal Chest for having an update of this guideline. And I also found out that there were some other individuals that were prominent in the community that were interested in such a guideline. And what I found, I was on the guideline panel in the College of Chest Physicians, and so I decided to write a proposal. And that's where this guideline started, is I wrote a proposal and we put it through a process. To get a topic developed, there are several things that are important. For one thing, it has to be something that's of general interest. It should be a topic that's somewhat controversial, because if you publish a guideline and everybody agrees before you've written it on what the answers are, there's no point in doing it. It won't get a lot of interest. There can't be any recent work, or not a lot of recent work. You want to kind of forge new grounds. And you want to look at the various societies and other stakeholders that are involved to see how important it is to them. One of the first tasks that I was involved with after we had the proposal approved was to select chairs. You want a chair who has international expertise and is very prominent in the area. You also want a chair that doesn't have conflict of interest, and that's really important because some of your panelists are going to have some degree of conflict of interest, and it really is up to the chair to oversee their work and to make sure that the document is unbiased. And so that becomes very important if you're going to pick your chairs properly. And so we picked some co-chairs that we thought would be good people who had a lot of expertise in this area to work on this process. Then you have to select your panelists. And what we usually do is go to the chairs and get lists of people that they think would be useful in developing the topic. And in fact, you begin even at that stage to start thinking about useful questions for the guideline, because you want to pick panelists that have expertise for those different questions. After you've selected a panel, then you do a literature search, which is very comprehensive. There are a couple of ways you can do a literature search. You can hire an evidence center to do a literature search. So in that case, you go to someone, a library such as at Duke or at McGill, who are used to doing these searches, and you empower them to do the search. They will charge you maybe $50,000 for a literature search if you use this. The advantage of doing that is that they're unbiased. They're not involved in your topic. They're just doing an unbiased literature search. The expense of that precludes most of us and most guideline developers from doing that. And so usually you have to find someone or some way to do this. And to give you an idea, the ERS is doing a guideline on sarcoid, And our librarians up in our library in K-17 are the ones doing the search. So that's a cheaper alternative that works, and that's what you do sometimes when you don't have other alternatives. After you get the articles, what you get is a list of hundreds or more of articles that pertain to your topic. And what you have to do at that point is, just by looking at title and abstract, you have to cull through those and pick out ones that may be relevant. You ultimately get those down to a certain usable volume. There are extraction tools that you use to extract data from specific randomized control studies. And then you do summative statistics on that information, doing meta-analysis and grading. There's a software that allows you to do meta-analysis called Review Manager. If you go to the Cochrane website, you can download Review Manager yourself for free. And it is the principal statistical software that you need to do a meta-analysis. So that's something worth knowing. And then when everything was finished, when everything is finished and you have the information, you grade the recommendation. There is a tool called the grade tool, which is used to grade how strong a recommendation is. And then on our guideline, we had a process of Delphi voting. What that means is that the person or the small group of people who are developing a recommendation develop it, write it. That is subjected to voting of the entire panel. If there are those in the panel who disagree with the way that the recommendation is worded, they give suggestions. The guideline or the recommendation is revised, and then you vote on it again in an iterative process until you get a recommendation that the entire panel, or at least 80% of the panel, agrees with. This is a more complicated slide of elements of the process. The reason that I put this up here is not to re-show you all of these things, but I wanted to point out this area here. When you get studies that you think are suitable for inclusion in the study, you have to do a number of things with these studies. You have to look for their validity, both internal and external, their study design. You want to look for their consistency and the precision of estimates, and the directness to the questions that you're looking at. And some of these studies may be rejected based on that. What you do is, there's a tool, and it's really nothing more than an Excel spreadsheet, but you use this spreadsheet to actually extract the results from studies. So if you have 3 clinical trials that are implicated in a question and all have the same outcomes, you take all of the information from each one of those studies, the total number of patients from each, the total number with certain outcomes, and you make a summative table using this extraction tool and develop what we call an evidence to recommendation summary table that helps guide what your recommendation is going to be. So here I have listed the initial CHEST guideline that came out in 2001. We decided that we had the resources to look at 6 questions for this particular guideline on mechanical ventilation. The co-chairs are here. Our panel are these individuals. Hopefully you guys recognize the name of some of them. I want to spend a second to talk about a PICO question. If you don't know what a PICO question is, you should. It's the basic core question that we use to develop guidelines and can be used for other purposes too. You want to identify the population, the intervention, the comparative group, and the outcome. And what's usually done is you develop a general question and then you try to boil it down to a PICO question that you can work with. So, next is a question for you. Which of the following are true about spontaneous breathing trials and ventilator liberation? Liberation success is improved by using inspiratory pressure augmentation during the spontaneous breathing trial compared to TPs. Frequency to tidal volume as a threshold metric, that is success less than 105 or failure over 105, has been validated for a spontaneous breathing trial with pressure support. Institution of protocols for spontaneous breathing trial does not improve liberation. And SIMV liberation strategies are as effective as spontaneous breathing trial strategies. And it's open for voting. This question implicates the first of our recommendations. Most of you said that B was true, and that means that most of you are wrong and probably need to look at our guideline recommendation. So based on the guideline, what we found was that liberation success is improved by using inspiratory pressure augmentation during SBT. And that's a better way, or was a more successful way of getting people off the ventilator than just using a spontaneous breathing trial with the TPs. The frequency to tidal volume is a useful measurement. And Martin Tobin's work in 1991 showed that the threshold of 105 was important. But Martin Tobin's work was done with a T-piece. If you go look at the original trial, that's how it was done. And reproduction of that work has never been done using CPAP or pressure augmentation. There have been some abstracts. There have been other people who have looked at it, but not in a comprehensive fashion. And so we don't know what the threshold is. It is almost certainly less than 105. I asked Martin Tobin himself what he thought, and he told me he didn't know. That was a couple of years ago. So if he doesn't know, I don't think I know either. And so it's not been really validated. The other thing that I think is important, especially for the fellows to take away, is that most of the experts are getting away from using the frequency to tidal volume ratio as the sole tool to decide whether somebody passes or doesn't pass a spontaneous breathing trial. What folks are trying to do is to use judgment and look more comprehensively at the trial itself. And that includes taking into account your impressions about how the patient is tolerating the trial. And that's very important. So I think that in the future you'll see more use of the idea that it's a judgment and not a number that you need to use. And the 105 has not been proven for CPAP. It's not something that's been used. Institution of protocols for SBT clearly improves outcomes. And SIMV and spontaneous breathing trials were compared in a 1995 Esteban study, and it was shown that spontaneous breathing trials were more effective. So our first PICO question was, in acutely hospitalized patients ventilated for more than 24 hours, should the spontaneous breathing trial be conducted with or without pressure augmentation? Should we do CPAP at 5 or 7 or 10, or should we do a T-piece? And these are our outcome meta-analysis data. On the left, you see a successful spontaneous breathing trial as an outcome. Outcome was extubation success, and on the bottom, you see short-term mortality. You can see that successful spontaneous breathing trial and extubation success, as measured by reintubation within 48 hours, were improved by using pressure augmentation. The number of studies that we used in this, there were 4 studies, 4 randomized control studies that we used for this. There were 2 that looked very specifically at patients who had an elevated PCO2, a respiratory acidosis, while they were on the ventilator. And these patients almost entirely had either heart failure or COPD as an underlying cause. And in those patients, in that smaller subgroup, not only were the successful spontaneous breathing trial and extubation success improved, but short and long-term mortality were improved as well. So particularly in those patients with a respiratory acidosis, using pressure augmentation, a CPAP mask, immediately after, or using spontaneous inspiratory pressure augmentation was good. So our first recommendation was for acutely hospitalized patients ventilated for more than 24 hours. We suggested that the initial spontaneous breathing trial be done with pressure augmentation rather than CPAP alone. One of the things about it is that there was a lot of variance or bias in the articles themselves. The data supported successful CPAP trials, and it supported the reintubation rate, but the other outcomes were less strong, and that's why it was a conditional recommendation. Are there any questions about that?
Where did the 5 to 8 number come from?
That came from the fact that different studies use different numbers. And you would be surprised, I think you would be surprised at the variance across the country at what is used. So we're used to using 7 and 5 for everybody, but that's not what's done everywhere. Different places use different pressure levels. Okay. One of the things that was done is that because of the way the studies were done and because of the way we looked at the studies, this only applies to the initial spontaneous breathing trial, the first one, and doesn't apply to the others. So one would think that the same thing would apply, but there aren't randomized controlled studies looking at subsequent spontaneous breathing trials. Okay, so the next question. Which of the following is true about the use of sedation? Dexmedetomidine has been shown to improve ventilator liberation success in multiple randomized controlled studies. Propofol infusion syndrome is associated with tachycardia, hypokalemia, and a metabolic alkalosis. An ICU sedation protocol that substitutes continuous fentanyl and propofol for intermittent midazolam will improve ventilator liberation. An ICU sedation protocol focused on minimizing sedation will reduce ICU length of stay. So everyone got it right this time, which is good. So Dex has been, there's some evidence that's interesting that say that maybe it's a useful thing, but the key here is the statement multiple randomized controlled studies. That's not been done. A couple of the better studies are patients serve as their own control, so they've tried first on the one and then the other. But there aren't multiple randomized controlled studies yet. B, everything is opposite of what it should be. So if you want to know about propofol infusion syndrome, just reverse all of that, and you'll be set. Intermittent sedation is better than continuous for getting people off the ventilator, and the sedation protocol focused on minimizing sedation reduces length of stay. Our question was phrased like this: in acutely hospitalized patients ventilated more than 24 hours, do protocols minimizing sedation compared to others impact duration of ventilation, ICU stay, and short-term mortality? Really hard to find good studies to prove this. We looked at some of this. You can see that the summary boxes are just touching or just favoring the sedation protocol, and that really has to do with the fact that the articles were quite different one from the other, and it was hard to get summative data. But there was some evidence that duration of mechanical ventilation, ICU length of stay were improved. So our recommendation was for acutely hospitalized patients. We suggested part of the grading of a recommendation is we use recommendation, the word recommendation, for a strong recommendation, and suggest for one that's less strong. So we suggested protocols attempting to minimize sedation.
Dr. Ouellette?
Yeah.
Minimizing sedation is explained as titrating to what, in the sense of
Yes.
fast, or is it talking about continuous or talking about intermittent?
Yes. Exactly. It means minimizing sedation. So the problem is when you look at the underlying studies that were used for this recommendation, and when you look at the literature, what you find is they're all being done different. So some are done trying to minimize continuous sedation, others are done intermittent versus continuous. They're all being done in a different way. And that's why the last statement, low quality of evidence, is there, because they're not being done in the same way. There's a lot of heterogeneity between the trials. There's actually some statistics you can use in the meta-analysis for that, but basically they came out as not being not high quality evidence just because of the reasons that you bring up. So there was insufficient evidence to recommend any one protocol over another, and what the value and preferences part on the guideline mean is sort of the things that you should consider. when you take a look at this, when we developed this recommendation, we placed a high value on reducing ventilator duration, and that was that influenced the recommendation being worded the way it was. So your next question, which of the following is true regarding the use of non-invasive ventilation post-extubation in a patient with COPD who has a chronic respiratory acidosis and has passed a spontaneous breathing trial? A, application of non-invasive ventilation should be delayed until the patient has evidence of respiratory failure post-extubation. Short-term mortality is reduced in high-risk patients immediately treated with NIV. Virtually all such patients can be treated with MIV with a face mask interface. The respiratory acidosis should be corrected before extubation is attempted. And your answer is what? Okay, I'm going to go to the answers. You guys said B, and you would be right. So short-term mortality is reduced in high-risk patients immediately treated with NIV post-extubation. Esteban did a really interesting study that was published 10 years ago. And it was one of the first studies looking at NIV post-extubation. And what they did was they had a group of patients who were extubated. When they failed extubation, when they started to develop respiratory, they took a subgroup that was failing and developing respiratory failure after extubation. And they took part of it, they randomized them at that point to either getting NIV or being treated with standard of care, which was usually to get reintubated right away. And what they found out was the NIV arm did worse. And because of that study, a lot of people were afraid, concerned about using NIV post-extubation. What the 4 studies that were looked at in this study, in this meta-analysis, what we found was that in all of them, they immediately applied NIV after extubation instead of waiting till respiratory failure developed. And so I think that this is an important point, and it's why I put the A response in there, is if you have somebody who you extubate, who you deem to be high risk, you should not wait until they start to have problems to use NIV. They should be immediately put on NIV after you extubate them. That's when you get the benefit of the good outcome. If you wait until they fail, then you may not be doing any benefits for the patient. The problem with NIV is you can't use it in everyone. There's lots of reasons why NIV may not work, because the face mask doesn't fit, or the patient vomits, or things like that, or they're not awake. There's lots of reasons why it may not be a good interface. The respiratory acidosis not only shouldn't be, but you usually can't correct it. They usually have a chronic respiratory acidosis, and you can't do anything practically about that, so you shouldn't correct that. So our question was, I think I've gone over most of this stuff right now. The Esteban article is listed there for you when you get this. There's lots of reasons why you might not be able to use a mask, and I've listed those things here as well. And so our PICO question here was, in high-risk patients getting mechanical ventilation for more than 24 hours who pass an SBT, does randomization to NIV versus not have a difference in these outcomes? And these are the forest plots of 4 of the outcomes. And the evidence was probably the strongest for this recommendation as for any of the ones that we looked at in this guideline. So they're pretty significant. So our recommendation was, for patients at high risk of extubation failure who have been receiving mechanical ventilation, we recommend preventative NIV. So we use the recommend word instead of the suggest, and the evidence was considered to be strong. So what is high risk? And that's a question. And the high risk, what was high risk varied a little bit between the 4 studies that we used. It seems that most of the patients who were high risk had, in the collective studies, had COPD, CHF, and certainly those patients with a respiratory acidosis while they're on the ventilator would be considered high risk. The important point again, NIV should be applied immediately post-extubation, and it may not be appropriate for certain patients. This is an important recommendation. I believe of all of our recommendations in this guideline, this is one that needs to influence practice.
Dr. Love,
Yeah.
the COPD and CHF, are they more comorbidities, or is it more of a patient with an exacerbation of both of those?
So as you might imagine, when you look at the 4 different trials, it's hard to tease that out exactly. What all of them had was verbiage or charts that indicated the reason for respiratory failure was, and then there was a group that had COPD checked and a group that were CHF was their main reason for being intubated. So that's where it comes from. There's obviously room here for judgment. So it doesn't mean that if you fall outside of that group that you're not high risk. I can imagine somebody with obesity hypoventilation syndrome who has a chronic respiratory acidosis who doesn't have either of those 2 illnesses who you would consider as high risk. That would be a common Henry Ford ICU patient. So there may well be others. It's not meant to limit to those things.
Dr. Ouellette,
Yes.
For how long do you keep those patients on non-invasive?
Good question. No one knows. So what I see in practice is that I think it's probably done for too short of a period of time. I've recommended using it, and 3 or 4 hours later I come back and the resident's taken them off. He was doing really well. I think probably 24 hours is what I would say. I think that's a reasonable recommendation, but the studies are different. There's heterogeneity, and so that answer is not really understood or known.
And Dr. Ouellette, the other question I have is, in all of these studies, when you put them all together, were you clear on what do you titrate the BiPAP for? Is it just patient comfort, or an endpoint of oxygenation or ventilation, as you see the tidal volume there?
The studies weren't done that way. The studies identified the patient and put them on NIV. And we're looking at durable outcomes such as I've shown you. And although they have some of that data in the articles, in the different articles, those were not the endpoints.
Did the articles specify like an average time frame that they ventilated them non-invasively?
They were each different, and I would have to look to give you that exact data. But it was usually 24 to 48 hours is the general time frame.
Dan?
Yes.
Given that time frame, have these studies been looked at in terms of expense, so the sort of a quality, how much expense there is related with how many lives you're going to, or re-intubations you're going to avoid?
It's a really good question. It's one that we talked about, and it's one that none of us knew the answer for. There were no economic analyses in there. And some of the things that we talked about is that certainly if you shorten duration of mechanical ventilation or ICU length of stay, you're going to improve costs dramatically. Mechanical ventilation is associated with high costs. On the other hand, knowing what we know, for example, from the Esteban study where people were randomized late and failed, that implied to us that during the NIV phase, these patients require increased monitoring and close observation. That may entail costs as well. And so we thought that cost may be an element and it's something important, but we had no way of examining that carefully. The next question. Which of the following is true concerning protocolized rehab programs for ventilation in the ICU, for ventilated patients in the ICU? These programs will reduce ICU mortality. These programs will improve patients' ability to ambulate on discharge. These programs will improve patients' independence on discharge. These programs will reduce ICU length of stay. It's open for you. Okay, finished. And the answer was they improve independence at discharge. They don't improve length of stay or the other outcomes either. So our fourth PICO question was, should acutely hospitalized patients who have been ventilated for 24 hours be subjected to rehab, which we do here. And here's the data. And of the outcomes that we looked at, mortality, ambulation at discharge, length of stay, and independence, the only one where the forest plot showed benefit was independence at discharge, and the others were not shown. And so my question was taken directly from our outcome summaries for those 4 outcomes. So our fourth recommendation was for acutely hospitalized patients who have been ventilated. We suggested a protocolized program. Again, the evidence was of low certainty, and it was a conditional recommendation. The evidence was not good. And we thought that there was insufficient evidence to propose one rehab program over another. And the values and preferences basically said, we think this is a great idea, but the outcome data is limited. Yes, sir?
So what if this is look at trained patients versus untrained patients?
Most of the if I remember correctly, the study did not involve trach patients predominantly. It involved patients who are there for primary ventilation, were intubated. Yes, sir. So, you know, thinking about the long-term chronic patient, I think it's a different patient population, and one would think that rehab would be very important. Okay. The next question, which of the following is most likely to result in successful ventilator liberation? Weekly teaching sessions for residents, targeting ventilator liberation, professional development courses for respiratory therapists concerning ventilator liberation, institution of a respiratory therapist-driven protocol, feedback to ICU physicians concerning their success rates. So, answer, you guys got it right. Protocols are good. So when you institute protocols for something like this, there's improved outcomes. So our fifth PICO question was, should hospitalized patients who have been ventilated be managed with a protocol for liberation or not? And this is a summary table of our data with looking at the odds ratios of the different outcomes. And based on that, we suggested that a ventilator liberation protocol should be used. But it was conditional, and there was low certainty of the evidence. So there were a couple of ways that these protocols have been inputted, either by computers or using checklists, but we didn't have we did not have sufficient evidence to recommend one or the other. And there was a high value placed on getting off the ventilator. And then, I'm not sure if this is the last or second to the last question, but this is a patient with COPD in your ICU who has been ventilated for 72 hours and is now ready for extubation. What is true about the cuff leak test? A quantitative cuff leak test should be performed in most patients, with a leak of 110 milliliters being more predictive of successful liberation. A qualitative cuff leak test should be performed in most patients, with no demonstrable leak being predictive of failed liberation. The cuff leak test is not predictive of weaning outcomes and should not be used. Systemic steroids should be administered for less than 24 hours to patients failing a cuff leak test. And your answer is what? And I'm going to check the answers. and kind of split between not thinking it's a useful test and thinking that it should be administered for less than 24 hours. The C answer is not really that wrong because there's not very much good data. And if you get experts in the room, they will argue about whether it should be done or not. So I wouldn't so much as say that is a wrong answer, is that I would say that D is probably a right answer and one that you should remember. So it's probably the best answer of the group. The problem with the cuff leak test is that there are multiple ways of doing it, and practically every article that you look at employs it in a different way. The qualitative cuff leak test means you let down the cuff and you listen for air, which is what we seem to do here, and that's not a very useful test. A quantitative test has been proposed with a leak of 110. There's some other metrics that are used. Most people don't use that. It's been used in some studies, but it's not clear what's the best thing to do. To look at, our first question was, should a cuff leak test be performed? And you'll see we had a lot of trouble finding data. So there were 14 observational studies. We had 2 prior meta-analyses. And I wasn't involved in this PICO question. What they actually did was they conducted something they called a simulated trial. They extracted the observational data and they tried to conduct a simulated trial using the data that was in the 14 observational studies. And in doing that, they realized that that was a little bit out there and probably not very solid data. And so they came up with the idea that if somebody was really sick and you thought that they might have a lot of problems with extubation, maybe you should do a cuff leak test. We suggested performing a test in a mechanically ventilated patient who meets extubation criteria and is at high risk. And what high risk means is anybody's guess. It's what you think is high risk. And how to do that test is really a question. What you should do is really not known. So it might be a good idea in certain patients. And I think that's the best that I could say there. But what happened was, in looking at that primary data, we also came across data about using steroids in patients who failed the cuff leak test. And we thought that the data that came up that we saw about that question was important enough to create a supplemental question. So our sub-question was, should systemic steroids, if you do that test and they fail, should you give systemic steroids? And there was actually some data that it helped. And so we found some randomized controlled studies that demonstrated that post-extubation stridor and reintubation rates were improved by giving steroids. So it's hard to give a good recommendation about doing the test, and it's really hard to give a good recommendation on how to do the test, but should you do it, and should they fail by whatever criteria you're using, they will probably do better if you give them steroids. So we suggested giving steroids for less than 24 hours prior to extubation for those who fail a test but are otherwise ready for extubation. And that's all I have. Yes.
Question. So
Questions?
after the steroids, do you
Yes.
check again the cuff leak or not, or just extubated patients?
The studies didn't show that. That's not what the studies did. Should you do that? That's a question of judgment. We could have a discussion about that, but I don't know what the right answer is. Yes, sir.
Yeah, and if we were gonna, one was gonna do that, right? So you would have a patient otherwise ready for extubation.